I'm feeling very honored. Our research on third international conference on advancement in electrical and electronic engineering, hosted by Duet. So myself, Mohammed Hassan Titu, and I'm the first author. So these are the topics we are going to cover today. I'm just moving forward as we have limitation of times. So first, the question comes: what is brain tumor? So brain tumors basically characterized by uncontrolled cell proliferation within the delicate human brain pose a formidable challenge in healthcare. So here comes the question: we are working on this topic. Basically, we are working on this topic because it is a major issue that's been basically faced by doctors. We have been discussed with many doctors, and they say that diagnosis of brain tumors from only MRI images is quite hard. And here we really thought that if we can really contribute a little so that the doctor can aid with, so that we really selected this topic. And our 3D Boolean processing extracts crucial features guiding medical professionals to integrate diagnostics. So basically there are two problems in existing methods. I really understand that the problems, that's why we really face these problems. So the two problems are existing methods for brain tumor classification and segmentation has been lacking with accuracy and efficiency. And the other problem is the need for a more customized approach in developing a 3D unit model arises to address specific complexities of brain tumor. Basically, as it's a very complex task. So we have read almost 20 to 30 papers, and here thus some simple approaches. The earlier approaches basically 2D unit, which is basically the prime version of unit, and also recent advances have been seen on CNN, which is convolutional neural network, and also there are really a little work in 3D convolution operations. So basically our objective is two. First, we wanted to enhance accuracy and efficiency, and the second one is we want to advance ourselves in quantitative and qualitative analysis. So this is the dataset description. We have collected our dataset from Kaggle. So these are MRI images. This one is flared, this one is T1, and the other two are T2 and T1 cortex. So here you can really see that this is the dataset where you can see non-enhancing tumor pool and also peritumoral edema, which is this one, and also enhancing tumor. So these are basically the tools by which we can see and really understand that whether a patient has brain. So this is our research methodology. So feed the data into our machine, and this is the input of our machine. Then we pre-process the data with many things because the data was not really in a nice format. So we applied ZSQ normalization and also resampling the intensity of our data so that the collections are basically well known and can be understood easily by the machine. And also we perform different kind of augmentation techniques. For example, we have flipped the data, we have rotated the data, we have also zoom in and zoom out the data, and we have also shrink the data. So then this is the training set. We have really divided the data into 3 sets so that it's really state distinguished. There are training set, validation set, and also testing set. And this is our basically 3D unit customized model. And we are going to talk about more about this in the next slide. And this is the final output you can see. So this is our 3D unit model. You can see that this is a customized model. It's not basically the original unit model. We have customized this, and there are total 7 layers here. You can see that first 4 layers are basically downsampling, and the other layers are basically upsampling. So the downsampling is also known as encoding, and the other layers are basically known as decoding. Even in each layer, we have performed 2 convolution layers. Here you can really see that, and also there is one max pooling layer, and the final layer is basically the group normalization layer. So we have performed this task in the first 4 downsampling layers. Then we have transposed the layer so that it can be decoded again and then perform the other task. But here one notable exception is in the final outcome. Here you can see that in final outcome we have uses 3D convolution with the softmax function because it's a classification problem. So that's why we used it. So here you can see that the final output of our model. Let's talk about experimental analysis of our model. So we have performed our model for 30 epochs, and you can see that our model performed pretty well in all the accuracy tasks. Like we have performed well in training loss and also in validation loss. Also we have seen a tremendous result in DICE score and validation score, also in Dakar scores. So this is the quantitative analysis. Here W2 means full tumor and TC means tumor core. So we have performed Dice scores and Jaccard scores in different sections, and we got our result here. You can clearly see it. So this is the confusion matrix of our problem. You can see that it's performed on the pixel levels, and our model really performed pretty well. You can really see that from this confusion matrix. Also, this is the accuracy matrix of our model. So we have performed different analysis, and accuracy matrix we have considered are precision, recall, F1 score, accuracy, and support. Here you can see that in identifying the background of the tumor and the whole model, our model performed pretty well. Also in the tumor section, our model performed exceptionally well. So this is the qualitative analysis. You can really see the output of our model, how clear it is, and it's basically 3D output. So I think that it will really help the doctors and other researchers to visualize the MRI problem of brain tumor. So this is the necrotic tumor core, which is a red line, and this is the peritoneal embedded tissue, and the others are basically enhancing tumor. So this is a pretty clear model. I think we can also develop this in the further. So this is the performance analysis of our model. Here we can really first say that no other researchers has ever performed Jaccard score analysis, and we are the first researchers who have performed Jaccard analysis, and we have got a tremendous good result. And we have performed also other analysis like whole tumor dice, tumor core dice, and enhancing tumor dice. And we performed pretty well also in enhancing tumor dice than any other researchers has previously done. So this is the result of our model. Here you can see that on the left hand side the actual